DNA replication is the process of copying and duplicating a DNA molecule in a semi-conservative way. In this way, a replicated double helix consists of one old strand of DNA and one newly made strand through the replication process. The two molecules that come out of DNA replication are complementary. They are separate but the same.
The first step in DNA replication is splitting the old DNA strand into what will be a leading and a lagging strand by transferring it through the helicase. As it goes through, the strands split into a leading and a lagging, differentiated between the replication fork, which makes it shaped like a V. You can differentiate the leading and the lagging strands because in our model, the leading strand has an arrow on the outside and the lagging does not. This goes to show that the third carbon in the actual structure will have OH, which all other nucleotides will bond to more easily. The process for a lagging strand will be more difficult.
As the leading strand passes through the DNA polymerase and each nucleotide passes through the active site, corresponding nucleotides are added to the strand, synthesizing it into a new DNA molecule. This process is continuous as the lagging strand becomes synthesized into another DNA molecule. In the lagging strand, the process of synthesis is backwards compared to the leading strand. As the nucleotides pass through the DNA polymerase, they reach a point where they can no longer fit. At that point, the molecules continue to move through the helicase until there is enough space for the polymerase to begin the process of synthesis again. The piece of DNA left behind is called the Okazaki fragment.
As you can see, in between the Okazaki fragments, there is a few leftover spaces. So the DNA ligase comes and fills in the spaces.
And that is how, through DNA replication, one molecule of DNA is converted into two molecules of DNA that are complementary and anti-parallel.